Good day, everyone! We will now discuss diversity and adaptations in Poriferans and Cnidarians. Poriferans, known as sponges, and Cnidarians as jellyfish, corals, sea anemones, and hydroids, are foundational groups in marine ecosystem, playing a pivotal role in maintaining biodiversity and ecological stability. As primary feeders, Poriferans contribute significantly to water quality and nutrient cycling. They extract microscopic organic particles from the water, recycling nutrients and supporting microbial communities. Additionally, their porous structures provide habitat and shelter for a wide variety of marine organisms, from microorganisms to small fish and invertebrates, fostering a complex micro-ecosystem. Here are the various threats that the Poriferans are facing. First is chemical pollution, such as industrial chemicals, heavy metals, and oil spill can damage sponge tissues or disrupt their microbial symbiosis. The rising of sea temperatures, where sponges are highly sensitive to temperature changes, which can disrupt their metabolic process and microbial communities. Next is ocean acidification. Reduced pH levels weaken the spicules of some sponges, making them more vulnerable to physical damage and predation. Next are pathogen and diseases. Sponges are susceptible to infection caused by bacteria and fungi, particularly in stressed environments where their immune system are compromised. Lastly is predation. Natural predators such as sea slugs, starfish, and some fish can contribute to sponge mortality, particularly in overpopulated predator environments. Examples of this are introduction of invasive species. Cnidarians, particularly reef-building corals, are indispensable in creating and maintaining coral reef ecosystem, often referred as the rainforest of the sea. Coral reefs harbor an immense diversity of species, serving as breeding, feeding, and nursery grounds for countless marine organisms. Furthermore, the symbiotic relationship between corals and photosynthetic zooxanthellae algae enables energy transfer and productivity in nutrient-poor tropical waters, supporting food chains and boosting marine biodiversity. These organisms are increasingly under the threat of various factors, such as coral bleaching. This occurs when elevated sea temperatures cause corals to expel their symbiotic zooxanthellae algae. This leaves corals starved of nutrients, turning them into white and making them more susceptible to death. Next is also ocean acidification, where acidic water reduces the availability of calcium carbonate, which is essential for the coral skeleton formation. Weak coral structures are more likely to collapse and fail to support their ecosystem. Next is overfishing and destructive fishing practices. Practices such as dynamite fishing or cyanide fishing physically destroys coral reefs and disrupt their ecosystem. Next is physical damage, such as anchor drops, boat collisions, and irresponsible tourism, such as stepping on coral reefs, can basically break corals or damage their delicate structures. Both Poriferans and Cnidarians also act as indicators of environmental health. Sponges can accumulate and reflect pollution levels in marine habitats, while corals are highly sensitive to changes in temperature, acidity, and pollution, making them clearly early warning systems for climate change impacts. The decline of these groups, whether due to ocean acidification, warming of waters, or pollution, can have cascading effects, disrupting entire ecosystem and threatening global biodiversity. Conserving Poriferans and Cnidarians require a multifaceted approach, combining habitat protection, climate action, pollution control, sustainable resource management. By implementing these measures, we can ensure the survival of these vital marine organisms and the ecosystem they support. So here are the ways that we can support our ecosystem and these organisms. First is we should establish a marine protected area where no fishing and no dredging zones to protected coral reef and sponge habitats. Next is coral reef restoration, where we should implement artificial reef structure and coral transplantation to restore damaged areas. Next is community-based conservation. It is really important to engage local communities in coral reef and sponge conservation efforts, to encourage the local divers and snorkelers to report bleaching events and sponge population changes. Next is enforcement of law and environmental laws. Strengthening marine conservation laws and penalties for illegal fishing and pollution is important so that everyone will be reminded of their actions.
At this part, we are going to discuss the key differences of Poriferans and Cnidarians. As what have been discussed earlier, Poriferans or sponges and Cnidarians, such as jellyfish, corals, and sea anemones. These aquatic organisms have unique structures and survival strategies that help them thrive in their environments. So here, we will be able to compare and contrast Poriferans and Cnidarians to better understand their specialized adaptations and analyze the ecological importance of these species. First, we are going to talk about their habitat and lifestyle. Poriferans or sponges are found in aquatic environments, mostly in the ocean. They are sessile, meaning they remain attached to a surface for their entire adult life. However, their larvae are free swimming, allowing them to disperse to a new locations. For Cnidarians, which include jellyfish, sea anemones, and corals, they are also mostly marine animals. But unlike sponges, Cnidarians can either be sessile, polyp stage, and motile, medusa stage, as what we call it. So some species like jellyfish can alternate between these two forms. That is during their life cycle. Secondly, we have here the symmetry. Sponges have an asymmetrical or sometimes radial body structure, meaning that their bodies don't have a specific shape or arranged in a circular pattern. In contrast of that one, Cnidarians exhibit radial or biradial symmetry, meaning again that their body parts are arranged around a central axis. This type of symmetry is useful for animals that don't actively chase prey or as it allows to interact with their environment from all directions. In terms of their body structure, sponges have a simple body structure. Their outer layer is made up of flattened cells that provide protection. So inside that cell, they have these choanocytes, specialized cells with flagella that help them move water through their body. The gelatinous middle layer, this is what we call the mesohyl. This contains the amebocytes, spicules, and protein fibers that provide the structural support. Cnidarians, on the other hand, are dipoblastic, meaning they have two main tissues layer, the epidermis, or what we call the outer layer, and the gastrodermis, the inner layer. This is separated by a jelly-like substance called mesoglea. Unlike sponges, they do not have choanocytes or spicules. Okay, next is their specialized cells. One of the biggest differences between Poriferans and Cnidarians is their specialized cells. Sponges rely on choanocytes to generate water currents and trap food particles. They also have amebocytes, which transport nutrients and help them structural components like spicules. Cnidarians, however, are known for their cnidocytes, stinging cells that contain nematocysts. These act like tiny harpoons, shooting out venom to capture prey or defend against predators. This adaptation makes Cnidarians effective hunters, yes, no? Despite their simple body structure. Next, water movement and feeding digestion. So, sponges do not have a digestive system. Instead, they rely on water flow system driven by what? By the flagella in their choanocytes. So this system allows them to filter feed, trapping tiny food particles from the water. Digestion occurs inside the cells through what we call phagocytosis, the intracellular digestion. And next for Cnidarians, they feed differently. They use their tentacles to capture prey and bring it into the gastrovascular cavity, where digestion begins externally before being completed inside their cells. And next, what we have here, let's talk about some examples, adaptations for survival as well. For examples, for these sponges, we have like Venus flower basket. And for Cnidaria, we have Hydra, sea anemones, jellyfish, and corals. We will also present some examples of these different sponges and Cnidarians. For Cnidarians, we are mainly focusing on the corals. So these examples or these specimens are found in our museum. Talking about their adaptations for survival, sponges, as we know, they have variety of species. So all of which rely on efficient water flow system to survive. Some freshwater sponges can form gymnos, special structures that help them survive in harsh conditions. For Cnidarians, their ability to alternate between sessile and motile forms allows them to thrive in different marine environments. Cnidocites provide both offense and defense, ensuring they can catch prey and protect themselves from predators. So that's how they adapt in their environment in order for them to survive. That's all for their key differences between Poriferans and Cnidarians.
Now, I'm going to discuss to you the definition of Poriferans. Poriferans, commonly known as sponges, belong to the phylum Porifera, which comes from the Latin term meaning pore-bearing. This name reflects their unique body structure, which is characterized by numerous pores and canals that allow water to flow through them. They are considered the simplest multicellular animals because they lack true tissues and organs, instead showcasing a cellular level of organization. Most poriferans are marine organisms living in saltwater environments. Some species can also be found in freshwater habitats. Now we will show you some examples of poriferans and their morphological features, as well as their structural adaptations and functional adaptations.
Euplectella aspergillum is a deep-sea sponge with a tubular, curved, and basket-like structure. It is found in deep-sea environments, often at depths exceeding 1,000 meters. Euplectella aspergillum is commonly distributed in the western Pacific Ocean, particularly near the Philippine Islands. Preserved specimens appear dirty white, however, in their natural habitat, they may be covered with fine deep-sea sediments. Euplectella aspergillum belongs to the group of glass sponges, with a siliceous skeleton that enables it to survive in deep-sea conditions where pressures are extreme. Its most remarkable feature is its external lattice-like skeleton composed of hexactin silica spicules, which interlock to form a strong yet lightweight structure. For its conservation status, the conservation status of Euplectella aspergillum has not yet been formally assessed. This sponge is a filter feeder, meaning it extracts microscopic organic particles from seawater using specialized cells called choanocytes. As water flows through its body, the sponge captures nutrients, providing an efficient method for feeding in nutrient-scarce deep-sea environments. Euplectella aspergillum has developed remarkable adaptations for deep-sea survival. Its silica spicules provide both rigidity and flexibility, allowing it to withstand deep-sea currents and extreme pressure. The cylindrical skeleton with helically spiraling ridges, on the other hand, enhances mechanical strength, preventing the sponge from collapsing under high-pressure conditions. For the sponge's functional adaptations, its lattice structure is designed to optimize mechanical support while minimizing material usage, making the skeleton both strong and lightweight. Another functional adaptation is its mutualistic relationship with shrimps. A pair of shrimp become trapped inside the sponge's rigid structure and spend their lives within, forming a symbiotic relationship where they clean the sponge while receiving shelter. This unique relationship has even inspired cultural symbolism in Japan, where Euplectella aspergillum is sometimes given as a wedding gift, symbolizing eternal partnership. These adaptations allow Euplectella aspergillum to thrive in deep-sea conditions where food is scarce and environmental pressures are immense. Its silica-based lattice structure ensures both durability and efficiency, while its association with shrimp highlights an important example of symbiosis in marine ecosystems.
Next species to the phylum Porifera is the Phylospongia foliescens. Phylospongia foliescens is the accepted name for the species Carteriospongia foliescens. As you can see, this sponge somewhat resembles the shape of an elephant's ear. For its morphological features, it has a thin-walled, whorled, or curved plate-like appearance that can also be described as a foliose cup-like sponge attached to the substratum by a short, hard stalk. It is said that it grows massively by subsequent generations, continuously growing from the notch of its peduncle. Phylospongia foliescens has a great white coloration when alive. In preservation, as we viewed it, it appears creamy grayish-brown in color, with a warty pattern along its surface. The noticeable external features of this sponge is the presence of oscula, openings that are raised above the surface bumps. These oscula are scattered irregularly at the surface of the sponge. The sponge also has numerous small pores called ostia. This species has no spicules present. In terms of its skeleton, it is composed of spongin fibers which are flexible and provide the sponge with structural integrity. This composition allows the sponge to adapt to the surrounding environment, particularly in strong water currents. For its habitat, Phylospongia foliescens is typically found in coral reef habitats, though it can also be found in other marine environments such as deep-sea regions. The coral reef is an ideal habitat because it provides a stable environment with abundant nutrients. For its distribution range, this species is widespread in the Indo-Pacific region, with its type locality in India. According to the International Union for Conservation of Nature, or IUCN, Phylospongia foliescens is not yet evaluated, meaning its conservation status has yet to be determined. For its feeding mechanism, as a filter feeder, the sponge relies on its porous structure, mainly oscula and ostia structures, to draw in water and trap food particles. The choanocytes or collar cells also create a current that helps bring water through the sponge where suspended particles such as plankton are captured and ingested. For its structural adaptations, Phylospongia foliescens has several adaptations that aid its survival. As mentioned earlier, its porous body structure, along with the presence of both oscula and ostia, allows water and the food it carries to flow through the sponge's internal canal system. The choanocytes which lie in the canals generate the necessary current and trap the food particles. The sponge dermis is also armored, often covered with foreign debris like sand, particularly sand nodules that compensate for the lack of spicules. which provides some protection against predators and additional support to the sponge to help it withstand strong water currents. For its functional adaptation, Phylospongia foliescens is a sessile organism that is attached to a substrate by its stalk. One of the most interesting functional adaptations of this sponge is its symbiosis with cyanobacterial symbionts. These bacteria perform photosynthesis, producing oxygen and organic compounds that benefit the sponge. In return, the sponge provides a suitable environment for the bacteria to live, creating a mutually beneficial relationship. Phylospongia foliescens also produces cytotoxic chemicals to deter predators and prevent them from feeding on the sponge by causing cellular damage or making the sponge unpalatable. These chemicals also serve as antimicrobial agents, protecting the sponge from infections and competing organisms by inhibiting their growth.
Next is the Calispongia aculeata, known as the branching vase sponge. It belongs to the group Porifera and it inhabits the coral reefs. Its shape and structure is like a vase-like structure with branching forms. Looks like a trumpet because of its tubular shape. The color of pattern of this is typically pink to purple with some variation shades of red, brown, and green. Its external structure, it has a large opening on top, which is the oscula, and the small spores on the surface of the water, which is the ostia. Its skeletal structure is collagen-based with flexible and fibrous mineral spicules. They are found throughout the tropical and subtropical waters of the Caribbean Sea, Florida, Bermuda, the Bahamas, and the western Atlantic Ocean. Its conservation status based on the IUCN is not yet assessed. They are filter feeders, which they trap food particles that they consume and expel the water that they filter. This feeding strategy allows efficiency to obtain nutrients from their environment while playing a crucial role of maintaining water quality. Their structural adaptation is that they have a large surface area which maximizes the water filtration, and it has a flexible structure for withstanding strong water currents. Its functional adaptation is that their symbiotic relationship with microorganisms like the cyanobacteria, which assist the nutrient acquisition. And they also have the ability to regenerate tissue after injury to maintain ecological role as a filter feeder.
Let's now move on to another group of organisms, which is the Cnidarians. These creatures are known for their unique adaptations and their vital role in aquatic ecosystems. From jellyfish to corals, they are some of the most recognizable marine animals. Let me start by defining what Cnidarians are. Cnidarians constitute a diverse phylum known as Cnidaria, which includes fascinating organisms such as jellyfish, sea anemones, corals, and hydras. The name Cnidaria is derived from the Greek word cnide, meaning nettle. This term refers to their unique stinging cells known as the cnidocytes, which are used for capturing prey and defense. One of the species in the phylum Cnidaria is Acropora cervicornis, commonly known as the staghorn coral. This species belongs to the class Anthozoa and order Scleractinia. It is a branching, stony coral characterized by its thick, upright branches that resemble the antlers of a stag. The morphological feature of Acropora cervicornis is that when it comes to its shape and structure, it has a distinctive branching structure with narrow, cylindrical, antler-like branches. Its color pattern is typically tan, light brown, or golden brown, often with white tips. In terms of their external structures, it has polyps, corallites, and tentacles. Polyps are small, tubular structures that extend from the branches and are equipped with tentacles arranged in multiples of 6. Corallites are skeletal cups that house the polyps and are angled towards the tips of the branches, and then the tentacles emerge at night, appearing as tiny finger-like structures that pump themselves up with water and resemble tiny stars scattered across a coral reef. The skeletal composition of Acropora cervicornis is calcareous, meaning it forms a hard skeleton made of calcium carbonate. This provides structural support and protection against predators and wave action. This species primarily inhabits coral reefs but also can be found in other marine environments such as intertidal zones and mangrove areas. Its distribution range includes Florida, the Bahamas, the Gulf of Mexico, and the Caribbean. Unfortunately, Acropora cervicornis is classified as critically endangered by the International Union for Conservation of Nature or IUCN. And in terms of feeding mechanism, this coral is both carnivorous and photosynthetic. It captures plankton using its tentacles equipped with stinging cells called nematocysts, while also relying on a symbiotic relationship with zooxanthellae, an algae that live within its tissues, to produce nutrients through photosynthesis.
For the second example, we have here Pavona frondifera, common English name, leaf coral, and this belongs to the taxonomic group of Cnidaria. For its morphological features, first, shape and structure. So, it is in the form of fronds, rounded and compact form, open and flat blade appearance, larger growth variance from 30 to 40 centimeter in diameter. So this coral has a unique foliaceous or leaf-like growth forms where its overlapping blades that maximize the surface area to capture sunlight. So as what you can see here in the picture. So for its color and pattern, it can be seen as pale or dark brown. Thirdly, its external structure. They have the presence of tentacles, polyps, and corallites that align in irregular shallow valleys roughly parallel to plate or front margins. Lastly, skeletal composition. Of course, they are made of hard coral, or let's say calcium carbonate. For ecological information, we have here their habitat. So they are mainly found in coral reefs and as well as deep sea. For distribution range, they occupy the Indo-Pacific area. Conservation status, so according to IUCN, they are least concern. For feeding mechanism, they exhibit photosynthetic and with symbionts. Lastly, we are going to talk about their structural adaptation. So they have this propeller-like morphology in large colonies. The open, curved blade structure improves water flow, aiding in the delivery of nutrients and removal of waste. Calcium carbonate skeleton provides structural support and protection for the coral polyps and predators and for physical change as well. Lastly, the fragmentation ability. So these colonies can reproduce asexually through fragmentation, allowing detached pieces to settle and grow into new colonies, aiding survival in adverse conditions. Functional adaptations. Symbiosis with zooxanthellae. The corals harbors photosynthetic algae, what we call zooxanthellae, within its tissues. Secondly, adaptation to water flow. The shape of the blades optimizes water circulation, ensuring oxygen and nutrient delivery while removing waste effectively. Lastly, for structural adaptation, we have competition strategy in the form of chemical defense mechanism. For example, employs the overgrowth strategies growing corals or algae.
Next to the group Cnidaria will be Lobophilia recta, or known as the brain coral. Its morphological feature, it has a massive dome shape with flat colonies along the meandering valleys and ridges. Its color or pattern ranges from brown, green, and gray to vibrant shades of red, orange, and often because of the fluorescing under UV light. Its external features: it has polyps, a fleshy tissue overlying a hard calcium carbonate skeleton. Its skeletal composition is a hard coral which composes a calcium carbonate. It inhabits the coral reef and often distributed along the Indo-Pacific region, including the Red Sea, Indian Ocean, and Western Pacific Ocean. Its conservation status based on the IUCN is vulnerable due to decreasing of population because of habitat degradation and climate change. And its feeding mechanism is carnivorous because they capture plankton by their tentacles and nematocysts, and they are photosynthetic because of the zooxanthellae. Its structural adaptation: it has a thick fleshy tissue for protection against predation and UV radiation. It also has an extensive polyp tentacles for capturing prey and defense. And also it has deep valleys to provide shelter for smaller marine organisms. Its functional adaptation: it has symbiosis with zooxanthellae for photosynthesis, providing energy and food for the coral, and it also has the ability to expand and contract polyps for feeding and protection. One of its functional adaptation also is the mucus secretion to remove sediment and deter predators.
Phylum scabra has a round and thick shape, with its structure varying from flat to highly arched. Live specimens are typically grayish-brown, with some showing pink or violet outer margins on the polyp. The external body is covered with polyps, which extend for feeding. Phylum scabra has a calcium carbonate skeleton, which provides a rigid structure and contributes to reef building. Phylum scabra is found in coral reef ecosystems throughout the Indo-West Pacific region. Its conservation status was assessed on July 11, 2022, and it is currently classified as least concern. Phylum scabra has a dual feeding strategy. It captures plankton and small prey using tentacles equipped with stinging cells called nematocysts. It also has a symbiotic relationship with zooxanthellae, photosynthetic algae that live within its tissues and provide energy through photosynthesis. To survive in the competitive coral reef environment, Phylum scabra has developed both structural and functional adaptations. Tentacles, equipped with nematocysts, allow the coral to capture prey and provide defense against predators. Septa, eternal skeletal ridges, provide support for the coral polyp and assist in digestion. It has a symbiotic relationship with zooxanthellae, which perform photosynthesis and supply essential nutrients to the coral. This allows Phylum scabra to survive in nutrient-poor waters. Its calcium carbonate skeleton helps form coral reefs, providing habitat for many marine organisms. These adaptations allow Phylum scabra to thrive in coral reef environments. By combining carnivorous feeding with symbiotic photosynthesis, it maximizes energy acquisition, ensuring long-term survival in nutrient-limited waters. Additionally, its calcium carbonate skeleton plays a vital role in reef building, supporting marine biodiversity by creating habitat for countless marine species.
For the last species of Cnidaria, we have Tubipora musica, or commonly known as the organ pipe coral. Now, let us dive into its morphological features. Organ pipe corals form colonies of deep red, thin tubes which is 0.2 cm in diameter and is connected by a horizontal plate. In life, white polyps with hollow tentacles extend from these tubes. So for its color and pattern, they are usually in bright red, pale, or dark brown. They also possess these external structures, such as spicules, polyps, oscula, and tentacles. And for their skeletal composition, they have this calcium carbonate, making the coral hard. For its ecological information, number one is habitat. They are mostly found in coral reefs and deep sea environments. For number two, its distribution range. So it is found in Indo-West Pacific, west to Red Sea, north to Japan, east to Vanuatu, south to Australia. And for its conservation status, Tubipora musica is classified as least concern by the IUCN 24-2, while rather not currently at risk, it remains vulnerable to climate change, ocean acidification, and habitat destruction. So conservation efforts are essential to protect coral reef and ecosystem. For number four, we have feeding mechanism. So they are usually photosynthetic with symbionts. For structural adaptations, tentacles with nematocysts capture prey and deter predators. A rigid calcium carbonate skeleton that withstand currents and damage. They also reproduce asexually, that is through budding, or sexually through gamete release. Lastly, for their functional adaptation, they have this symbiosis with zooxanthellae. What does it mean? So, this forms a symbiotic relationship with zooxanthellae, which provide essential nutrients through photosynthesis. So, that's it for the species of Cnidarians.
To sum it up, Phylum Porifera and Cnidaria species have unique skeletal and morphological features that support their feeding, protection, reproduction, and overall survival. Poriferans or sponges are pore-bearing animals with a pore structure that allows water to flow through their bodies, filtering out food particles. This filtration system helps them thrive in nutrient-poor environments. While Cnidarians such as corals, jellyfish, and sea anemones possess stinging cells called cnidocytes, which are used for capturing prey and defending against predators. Both groups form symbiotic relationships with photosynthetic organisms. These organisms provide essential nutrients through photosynthesis, allowing sponges and cnidarians to survive and flourish in environments where food is limited. Sponges and cnidarians face multiple threats, including chemical pollutants, rising sea temperatures, ocean acidification, infections, and predation. These pressures compromise their health and survival, disrupting vital ecological functions. In this exercise, the conservation statuses of most species being assessed are mostly categorized as least concern and not yet assessed or evaluated, but there is also vulnerable and critically endangered species. To protect these sponges, cnidarians, and other marine species, there is a need for a multifaceted conservation approach. These key functions include establishing marine protected areas or MPAs, restoring coral reefs, engaging local communities, supporting citizen science, and enforcing environmental laws. In conclusion, a holistic strategy that combines habitat protection, climate action, and sustainable management is essential to safeguarding sponges and cnidarians and maintaining healthy marine ecosystems.